Possibilities of analysis and correction in the KME system. An extensive set of markers is used to analyze the body's condition in the KME system. A marker should be understood as a digitized and stored in the system the average statistical form of the electric field of various physiological process or substance. Markers are combined into different databases, according to the principle of belonging to a particular group, with similar physiological functions or properties. Combining marker bases into folders of different levels is also carried out on the principle of physiological and functional similarity of their action. The set of markers used in the KME system allows for assessing the condition of the body's physiological processes at different hierarchical levels of its organization, from the level of functional systems, organs, tissues, and cells, to the level of molecules. The markers bases are common both for auto-analysis, carried out automatically by the ERIQ Life software according to certain algorithms, and for independent, manual analysis, according to the system user's algorithms. The first in the list of marker bases is the System Indicators database. This database allows to evaluate the dynamics of homeostasis of the main functional systems, such as the immune system, endocrine system, nervous, cardiovascular, gastrointestinal tract, respiratory, genitourinary, and other body systems, and also allows us to evaluate some systemic integrative indicators. The second position in this list is the folder Organ Tissues. This folder allows us to determine which tissues and organs make their maximum contribution to changing the normal homeostasis of a particular functional system. The KME system allows us to conduct a profound analysis of the condition of functional systems and organs, up to the level of functioning of cells and molecules. Folder General Tissue Characteristics. In the folder General Tissue Characteristics, one of the essential characteristics of biological tissues is the condition of the tissue's extracellular matrix, ECM. ECM is a complex molecular system in which the cells of our organs and tissues live and function. Structural and functional changes in the matrix can lead to significant changes in the functioning of the tissue of a particular organ. It is known that there are no diseases that would not cause structural and functional changes in the ECM. We can observe and evaluate the condition and functioning of the extracellular matrix, which provides such functions as nutrition of the organ tissue cells, the excretory function of the ECM, and the function of intercellular communication. It should be noted that in the course of analyzing the condition of the processes, we can select and put to compensation markers that deviate from the physiological norm. The system allows us to determine the signs of toxic load and the functional orientation of the action of these toxins. Folder Hormonal Regulation. A significant proportion of metabolic and regulatory processes in the body is carried out with the help of hormonal liquid media, such as blood and lymph. The hormonal regulation folder contains the primary markers, by analyzing which we can assess the nature and level of hormonal regulation. These markers include blood elements, are the main blood cells, enzymes, hormones, and molecular factors that affect the metabolism of fatty acids in the body. Assessing the functional condition of the tissue of any organ, one can analyze the ratio of the balance of pro-inflammatory and anti-inflammatory responses of the immune system. This analysis allows for assessing the presence or absence of inflammatory processes in the tissue of an organ, as well as the direction and degree of activity of these processes. A feature of this folder is the ability to determine the body's resources to maintain normal homeostasis and normal immune status. It is important to note that the functional activity of various neuronal structures of the brain and the regulatory molecules produced by them, such as neuromodulators, neurotransmitters, and neuropeptides, has an impact on the body's immune status. The folder Cellular Metabolism contains databases of markers that enable to evaluate intracellular metabolic processes. A vital characteristic of any tissue metabolism is the activity of mitochondria, which produce the primary energy substance, the ATP molecule. With pathologies and aging of the body, the number of mitochondria and their activity in cells decreases. The amount of ATP produced also decreases. Working with the markers of the mitochondrial metabolism database not only allows us to evaluate the activity of mitochondria, but also provides important recommendations for activating their function and consequently, for normalizing tissue metabolism. The cellular metabolism folder contains databases of markers for two important, one might even say, key processes, maintaining the balance of which in the body is essential. These are autophagy and apoptosis. Autophagy is an intracellular process that frees the cell from non-functional intracellular structures. Disruption of cellular autophagy can lead to cell aging. Apoptosis is a regulated, genetically determined process of removing unwanted cells that the body, for various reasons, wants to get rid of. Cells of the immune system, phagocytes, take an active part in this process. The folder Neuronal Regulation combines three subfolders, such as brain structures, autonomic regulation, neural regulators. Assessment of the condition of brain structures allows us to determine and balance many specific features of the human body, and their psychosomatic orientation. Some brain structures are involved in the regulation of the autonomic nervous system, sympathetic and parasympathetic. The autonomic nervous system, in turn, innervates the entire body, all organs and tissues. The balance of the sympathetic and parasympathetic nervous systems is important for both mental and physical health of a person. The database muscular innervation and the spine condition largely depend on the balance of the sympathetic and parasympathetic nervous systems. The folder Neuronal Regulators contains databases of markers that combine the main molecules, neuropeptides, neurotransmitters and neuromodulators that determine the functional activity of brain structures, the autonomic nervous system, muscle innervation, and the spine. In particular, markers of neuropeptides and their activity make it possible to assess the ability of the integrative systems of our body to regenerate, self-regulate, and maintain normal homeostasis of many other functional and physiological processes. Folder Psychosomatic Factors. The bases of markers in this folder allow us to analyze the most relevant psycho-emotional components of a person's condition at the moment. The database-induced psychological programs lists the behavioral personality traits that have been formed as psychological characteristics and can manifest themselves with relative stability and periodicity. The folder Correction contains bases of markers, trace elements, vitamins, drainages, muscular activity, and food products. Based on the analysis results, it is possible to recommend appropriate microelements and vitamins for the course intake. The analysis of the drainages database focuses on those functional systems and tissues of organs whose own drainage or detoxification resources are insufficient for their purification, excretion of endogenous and exogenous metabolites and their toxins, and a general decrease in the level of tissue homotoxicosis. The muscular activity folder can be useful for analyze excessive muscle tension and accordingly, requiring relaxation, and insufficient muscle activity, the study of which will increase their tone. The bases of the muscles of the head, face, and neck are not only an emphasis on maintaining beauty, but also an indication of the possible causes of an imbalance in the autonomic nervous system. The last folder, Foods, allows us to give recommendations on specific foods that slow down metabolism and are usually not desirable in the diet, as well as on several foods that support the essential metabolic functions of organ tissue and functional systems. Markers of all bases, available in the KME analytical system, are used not only for the actual assessment of the condition of the human body, but if necessary, can be selected to compensate them, and therefore to correct the functional orientation of their action, in particular processes of the human body.